And the recording has started. Thank you. Well, I'm going to begin with going over our speakers tonight. I am going to start. I'm Priscilla Crawford. I'm a conservation specialist at the Oklahoma Biological Survey, and I'm going to start with a really brief introduction to phenology and also how climate change is affecting phenology and the life cycles of plants and animals in our ecosystems. Our second presentation is my colleague, Dr. Abigail Moore, who is a curator at the Bebb Herbarium, and she is also a botanist on campus and works also at the Biological Survey. And then our third speaker will be Claudia Gauss, who is our student research assistant at the Biological Survey. She's a biology major at OU, and she will be talking about our community science project, Redbud Buddies, which is a way for you to report phenology data about our state tree. So I will get started with our introduction to phenology. I am a conservation specialist. I do things across the state related to rare plants and animals, and also invasive species are my expertise. So phenology is not something that I have done direct research on, but as an ecologist, I have learned quite a bit about it over the last several years. So what is phenology? So phenology is studying the cycles of plants and animals and the natural phenomenon as it relates to climate and the physical environment, usually related to seasons and the cycles that we see in our ecosystems. You can kind of think of it as nature's calendar. We all have this, we all understand phenology in a very broad sense. So as most of us who have grown up in the Northern Hemisphere in a temperate ecosystem, we understand these cycles kind of in the back of our mind. We know that the leaves turn colors and fall off in the fall. We know that birds migrate south in the winter and come back in the spring. We understand these things on a very basic level, and phenology, the science of phenology, is studying that in more detail and getting precise measurements of these cycles and how temperature and precipitation, the climate, and day length affect these life cycles of the flora and fauna that we have in our habitats. So studying phenology is relatively simple. You simply record dates of that these life cycle events happen. So right now, here we are in spring. We are starting to see migrating birds come back. The birds that breed here in the summer are returning from their southern wintering habitats. We also see that the leaves are starting to break out of their buds. We see flowers start to break out of their buds. Speaking of our Oklahoma State tree, I looked at the one in my front yard, and the buds of the flowers have just broken out of their bud scales, and they haven't opened, but they're little bitty about to open flower buds. So all of these days in spring can be recorded, and we can study these life cycle events. But it's not just spring. Like I said, things that happen in the fall, things that happen in the winter, like leaf dropping and leaf color change and fruit ripening, and things that happen throughout the winter too can also be studied through this phenology. So it's simple data, but it takes years to accumulate for us to be able to say anything about the patterns that we see, because you can't just take data in 1 year and think that you understand the pattern, because there could be anomalies in that year. And so you need lots of years of data to average out to understand kind of the overall picture. Here are two pictures of field notebooks that were written 100 years ago that help us understand better phenology that was going on at 100 years ago. And these notes can be compared to what we observe today. Oh, I'm going to go on a slight tangent here. Kind of related to these two pictures of the field notebooks. I found these two pictures on the website for the Biodiversity Heritage Library, which is a program that the Smithsonian's part of. You can find a bunch of records for this kind of historic data and kind of history of science on this website, biodiversitylibrary.org. And the Field Notes project was what was on that previous slide. And it's a fantastic resource. So if you want to totally nerd out on historic data about natural history or science, this is a really cool resource. And you can kind of go down a huge history rabbit hole. And this month, they are featuring women in natural history because it's Women's History Month. And this picture that is on the slide now is women who were botanical illustrators. And these are some of the plates that they did for botanical illustration. So it's a cool resource if you're interested. Okay, tangent over and back to phenology. So why, there are lots of reasons why people need to understand phenology, not only because it's good to understand the natural world around us, but it's really important for things like agriculture. Ever since we have been doing agriculture, we have needed to understand the life cycle of the plants and the animals that we have domesticated. So when do we plant stuff? When do we harvest things? And we find examples of people keeping track of the seasons through things like Stonehenge and lots of other ancient structures that kept track of seasons to help them better understand when it was going to be a good time to plant or harvest, or when pests were going to be a problem. The life cycle of pests is an issue with agriculture. So agriculture is important, and we need to understand phenology for it. Also for health. A lot of you probably have pollen allergies, and you look at those pollen reports, and it's a good idea to understand the life cycle of the plants that are producing all that pollen out into our world. So understanding the phenology of the cedar trees and the ragweeds and the flowering trees in the spring that produce all these allergens is important. It's also important to understand the life cycle of pests and the disease vectors like mosquitoes that bring in diseases to us. So we need to know what triggers those life cycles for them to emerge and to come into our environment. And, oh, sorry. Also, in my line of work, it's important for us to understand phenology if we want to do any conservation or restoration of species or habitats. So we need to understand when insects emerge. If we want to protect those insects, we need to understand what they need to survive. What are their host plants? In the case of the monarch butterfly, we need to understand what kind of food sources things will eat, and when they are available to do the conservation work that we do. So by now you probably, and I've hinted at it, of course, in the title, phenology is really affected by climate and consequently climate change. And phenology is a way that people have been able to study how quickly the climate is changing in relationship to human activity. So we can go out and measure how warm the atmosphere is and how much warmer the oceans are and how much ice is melting from glaciers. But one of the other measurements that we do for climate change is looking at the change in plants and animals over the time that humans have been adding CO2 to the atmosphere. So it's been well documented that phenology is shifting with respect to climate change and human-induced climate change over the last 100 years. For example, in our neck of the woods, the Northern Hemisphere, and the ecosystems that we inhabit here in Oklahoma and the United States and the Northern Hemisphere in general, for every decade, spring has come earlier 3 days earlier. So for every 10 years, spring has advanced for 3 days. I'm almost 50 years old, so that's 5 decades. 3 days times 5 is 15. So in my lifetime, spring has advanced over 2 weeks. So spring is earlier by 2 weeks since I've been alive. And they have been able to study these things by looking at the flowering times of plants, egg laying and the migration of birds, emergence dates of butterflies and hibernating animals, and also looking at satellite images and looking at the green up of vegetation over the course of the last several decades. So one of the problems that is popping up with this shift in like earlier springtime and things staying warmer later in the season is that we get something called phenology mismatch related to climate change. Because as you are aware, we have ecosystems full of species that are interacting with one another. One species will eat another, one species will pollinate another, and these interactions can be affected by climate change because individual species may have a different phenological driver. So a bird may be affected by day length, which isn't changing with climate change, but the insects that it eats may be affected by temperature, which is changing. So these interactions between these two species may start to shift because they are being affected differently during these changes in the climate. So I'm going to go through kind of a really simple example, a very, very simple example or simplified example of this. So for example, an oak tree. I'm going to talk about 3 species, and we're going to start with oak tree. So an oak tree, say this is a post oak, Quercus stellata. It needs light, it needs water, it needs air, so carbon dioxide and oxygen, and it also needs nutrients to grow. But things that trigger its life cycle, the phenological or phenology drivers, are things like temperature, day length, chilling period. So chilling period kind of means like how long is winter. Pollination. So when does the pollen leave the flowers and then pollinate other flowers to create a new seed, and then when are those seeds dispersed? So these are some of the phenological drivers of an oak tree. Then you have a caterpillar that eats oak leaves. So again, it needs air, food, and shelter. And its phenological drivers are temperature and then the presence of young oak leaves. It needs the young, tender oak leaves to eat so that they can then grow and then eventually pupate into a moth. And then we have the eastern phoebe, our songbird. Again, it needs air, food, and shelter, but its phenological drivers are day length. That's what triggers it to migrate and to build its nest and lay its eggs. But then for that life cycle of the nestling to continue, it also needs a lot of protein for that offspring. So let's put these 3 species together in kind of a simple interaction. So we have our eastern phoebe arriving from migration. It arrived based on it's triggered by day length. It lays its eggs, and then the eggs hatch, and the nestlings are looking for caterpillars to eat. Currently, the situation is that the caterpillars are highly abundant during this period where the nestlings need a high protein diet. So these 2 populations are kind of matched at this point. But what happens when we have climate change? In this very simple example, we could imagine that the bird continues on the same timeline, arriving because of a certain day length, laying its eggs hatch, and then the birds are available. But if the caterpillar is emerging earlier because of a warmer temperature, it's emerging back here during a different part of the cycle, potentially when the eggs are being incubated. So this peak in caterpillars is happening before the nestlings are available to eat, or before the nestlings have hatched. So at this point, there are few caterpillars. So this mismatch in interaction could happen if the interactions, if the different species are affected by different phenological drivers. So that's a concern because all these nestlings of phoebes won't have caterpillars to eat. Now, this is an extremely simple example. As I'm sure you know, the ecosystems are based on dozens and dozens of species that have multiple interactions. Phoebes don't necessarily eat one species of caterpillars, and then the caterpillars may be driven by something else, and the phoebes may adjust. Their behavior may be plastic or adjustable so that they figure out in some way that they need to lay their eggs later, or pardon me, earlier. They need to arrive earlier, or maybe maybe we don't understand their phenological driver and it's not really day length. It's something else, maybe related to the climate on their wintering grounds. So I just want to throw that caveat out there, is that things are much more complicated than I'm portraying here in this little graph, or that has been portrayed by these researchers in this little graph. But it is one of the issues that people are concerned about related to climate change, that we'll start to see these instabilities and interactions between species. So some of the other things that could potentially be happening between that are mismatches. That so we had the example of the oak, the caterpillar, and the phoebe, but it could be that we have an asynchrony between plants and their pollinators, which is more likely to happen with species that are really specific. So like a certain bee will pollinate a certain plant. And not so much of a problem if there are generalist species out there. There may be some interactions that will be affected by, because herbivores emerge earlier from their hibernation than animals that are kind of higher up in the food chain. Excuse me, I'm going to take a drink of water. So these ecosystem functions and the community structures that we see in these complex ecosystems will definitely be affected by climate change, and phenology will be affected, but we don't understand how that's going to happen in a simplified way. So just know that things are going to change, but ecosystems have been changing forever, since ecosystems came together. So we even see some of the ecosystems, like in the eastern part of the United States, still responding to the glaciation that happened 10,000 years ago. So these changes are always occurring within ecosystems. Ecosystems aren't static. We kind of try to think about them that way because our small human brains, it's easier for us to understand, but ecosystems are always changing. The concern here with climate change is that we're causing the change, and it's very quick. And by creating these unstable ecosystems, it also makes human populations unstable. And another feature related to phenology and climate change is that climate has become more variable. And some of the species that we have are not well adapted to this variation. So most of you who have been living in Oklahoma for the past year will remember last winter's deep freeze, what I like to refer to as the deep freeze. So at the end of last winter, we had several days of extremely low temperatures, extremely low for Oklahoma. And this caused kind of a crisis with a lot of the species that were in Oklahoma during that time. Food became extremely scarce. Water was frozen, so things that relied on open water were having troubles. So, but we had had warm periods before that. So if something emerged earlier, if we have these late cold spells, a lot of species aren't well adapted to this extreme variation. So, for example, plants can be damaged, and then insects or can emerge early, but then have these cold snaps kill off those early emerged things. So a lot of the plants and animals that we have in our state may not be well adapted to these climate extremes and climate variability. So with that, that was an extremely fast overview of a very large science and a body of research. And I kind of like rushed through some of the climate change information. I don't see any questions in the chat. If somebody wants to throw one up there real quick, maybe it was too overwhelming, and I hope most of you were able to kind of follow the basic premise of how climate change is affecting phenology. But if you think of a question, feel free to pop it in the chat when you think of it, and we can answer it at the end. Otherwise, I guess I'll pass it over to Abby, who will take over and talk about how they are using herbarium specimens to do research on phenology.
Sounds good. Thank you, Priscilla.
You're welcome. Let's see. Are you able, do I need to switch the screen, or are you able to take over?
I think I can. Let's see. Okay. So yes, I want to talk about using herbaria to look at phenology in plants. And the first question is, what is an herbarium? So I am the curator of the Robert Bebb Herbarium, which is the herbarium at OU. And it is a collection or an archive of dried pressed plant specimens. So if you go into the herbarium, you just see a bunch of cabinets, because this is where the plants are kept. And then you can open the cabinet and you see folders of herbarium specimens. So you can open a folder. It has the name of the species on the outside, Juniperus monosperma. And then when you open it, what you see is cardstock with these dried pressed plant specimens glued to it. So these are a record of plants that existed throughout Oklahoma, throughout the world, and what they were like at a certain point in time. So we can look at an individual herbarium specimen, and obviously the first thing that you see is just the pressed plant. And we can see at this point that this plant has the flowers that are blue, but it also has a bunch of developing fruits. So it's at a particular life stage. And then we can look at the label and see when and where it was collected. And this means if this plant was collected on June 13th, 1958, in Haskell County, we can see that this plant, Delphinium carolinianum, one of our larkspurs, was both flowering and fruiting on that day at that place. So this is a record of phenology, even if the person who collected it in 1958 or the people who collected it weren't specifically making a record of phenology. Maybe they were just collected it so they could identify it. A couple of other parts of the herbarium specimen. We have our symbol. We have a couple different symbols on the herbarium sheets and number, so we can keep track of the specimens and they won't get mixed up with specimens from other herbaria. And then we have a barcode that we put on all of our plants as we are imaging them. And then we also have these annotation labels, which talk about if the plant was included in a study or if the name of the plant was changed by someone. And then at the top, which I didn't label, these two things, the ruler and the color scale, those are just for those aren't part of the specimen itself, but those are included in the images of the specimen so that we know how big it is and what the true colors are. So just a small overview of the herbarium. It was refounded in 1903 because we had an herbarium on campus before then, but it was destroyed completely in a fire in 1903. So our existing collection dates from 1903. And it contains over 250,000 specimens. So we don't know exactly how many because although they have numbers, sometimes the numbers have been duplicated or potentially some of them have been skipped, and we haven't counted them all because they're not all in the database yet. Of these specimens, about half are from Oklahoma, and the rest of them are from a lot of them are from the surrounding states, and then kind of progressively fewer as we go farther from the state, but basically throughout the whole world. So our oldest specimens are from 1894. So of course, these are specimens that were not part of the original collection, but were gained either as gifts or in exchange for our own specimens after 1903. And the newest is from last year because we are still actively collecting to document the flora of Oklahoma and other places that we are doing research. And what is exciting is that we're currently imaging all of our specimens and putting them online. So we have, we're part of the Torch collection, the Texas-Oklahoma Regional Consortium of Herbaria. And this is an online database where you can find information about plants from a whole bunch of different herbaria, as well as images. And this is just a search for Delphinium carolinianum, the specimen that was in the picture where I labeled all the parts of it from Oklahoma. And so these are not ours, these are herbaria of some of our colleagues in Torch, but our specimens are further down as you scroll down because they're in alphabetical order. But anyone can access this and get a lot of information about plants from Oklahoma and indeed throughout the world. So if you just Google Torch Herbarium database or Torch Herbarium portal, then you will be able to find this. And here's just an example of one of these old specimens from 1894, plants of Indian Territory. This little plant looks kind of like a shamrock, but it's actually a fern that's just a very unusual fern that is found various places in Oklahoma. So we have phenology. So as Priscilla said, you can look at the timing of a whole bunch of different life history events. And a lot of these are actually things that you can see on herbarium specimens. So you can document flowers and fruits and buds. Sorry, my cat is in the background complaining. You can see when new leaves are formed, when leaves are being dropped in some cases. So there's a lot of phenological information that you can get from herbarium specimens. And of course, the huge advantage that herbarium specimens have is that you can look at these field notebooks and they record one person's observations at one particular time. But the herbarium has this history of 125 years of plants from throughout the state of Oklahoma. And so we have a record of phenology over that entire time. So there are quite a few studies that people have done on phenology using herbarium specimens. So this is from a study of herbarium specimens from the Rocky Mountains in Colorado. And this is just an example. We have two different species. So the ones on top are Leucocrinum montanum, the sand lily, and the ones below are Pulsatilla patens, the pasque flower. And we also have two different kind of time periods. On the left, we have older specimens, and on the right are younger specimens. And you can see that the older specimens were collected much later in the year, around the end of May. while the younger specimens of the same plants from presumably approximately the same place were collected much earlier in the year in April. So we have this difference of about a month between these collection dates. And this was true from throughout their study. So we have a documentation of this change in timing of flowering. And they could gather the data from a lot of different specimens and graph it. And I will be showing a lot of similar plots of data like this. So the horizontal axis in this case is year, and we always go from older to younger. There are a couple of different, sometimes we have temperatures, so I'll show that when we get to it. And then the vertical axis is flowering date. So the earlier dates earlier in the year are lower down, and the later dates are higher up on this axis. And this is probably not a way that you're used to looking at the dates, but this is just makes it easier than listing the actual month and day to just number the days of the year consecutively from 1 on January 1st to 365 on December 31st. And so that way we don't have breaks to deal with. And since we're in the Northern Hemisphere, it is easy because most of these plants aren't doing anything actually in the winter. Obviously, there's ways to get around it. So each of these little symbols represents one herbarium specimen, and the different colors and shapes are referred to the different species with these kind of cryptic abbreviations that are listed over here in this column. And they have, the graph has trend lines showing the overall pattern shown by these herbarium specimens. And in this case, these lines slope downwards, and this means that flowering has become earlier over time. So it used to flower in May, now flowers in April, and so forth. And in fact, when you look at all of the data, the flowering date for these species has gotten about 3.2 days earlier each decade, or 42 days earlier over the whole time period. Yes, it became too distracting. She had to come up. So then we can look at a different study that is herbarium specimens in the southeastern US, where the pattern is a little more complicated. So here, again, we have our day when the plants flowered on the vertical axis. This time these flower maybe earlier in the year, so it's 0 to 150. And then on the horizontal axis, we have, instead of the year, we have what they call March temperature deviation. And so this is just how cold or warm it is, because as Priscilla said, instead of just warming, climate is also becoming more variable. And so we're getting sometimes really cold winters, sometimes really warm winters. And so this makes it, we can see more precisely what's going on if we look at the specific temperature. So we have a bunch of gray lines, which represent the trends for individual plant species. And then the green dotted line is the overall trend across plant species. So we can see, again, like in the previous chart from Colorado, the lines slope downwards. And this means that flowering is earlier in these spring flowering plants when March is warmer. So just like we would expect. But you think, well, spring flowering plants are going to be very strongly affected by March temperatures because that's when they're flowering. So it really matters a lot. But what happens when you are looking at summer flowering plants? How are they affected by March temperatures? And so this is the same study, but instead of spring flowering, these are summer and fall flowering plants. So again, there were a lot more species here. So we have our horizontal lines, each represent a different species. And the green line is the overall trend across species. And here you can see that the relationship is much weaker. The lines are close to flat. So this means that the flowering of these summer and fall flowering plants is not really affected by the March temperature that much because they've grown, they've had a lot of time since then to be affected by other things. But then we can look at how these fall flowering plants are affected by the July temperature, which is much closer to the time that they flower. And in this case, you can see that the line actually slopes upwards slightly. So this means flowering is later when you have a warmer July. And I guess this can make sense. Maybe July is, if it's warm, maybe they're more drought stressed, so they're not growing as well. So they need more time to grow before they can flower. So especially when you get later in the year, the response to warming can be quite complex. Sometimes it can make things earlier, sometimes later. And so you have to look at a bunch of different things. And now I want to talk about some undergraduate research that was done by a student in the herbarium, actually his honors project, John Unterschutz. And he looked at 4 different plants in the sunflower family. And each of these plants had over 100 specimens in our herbarium. And so he could look at the phenology of these plants very closely because we had a lot of specimens. So our 4 contestants are Grandelia ciliata, or Spanish gold, Liatris punctata, or blazing star, Retibita columnifera, or coneflower, and Vernonia baldwini, or ironweed. And these 4 species would all fall into the summer to fall category. None of them are spring flowering plants. And I'm going to specifically focus on Retibita columnifera, the coneflower, because each of them are slightly different and it would be too much to talk about all of them. So instead of just classifying the specimens as flowering or not flowering, he divided them into phases depending on how many of the flowers were open. So they could be just starting to flower, they could be full flowers or peak flowering, they could have just be at the end of their flowering or last flowering, or flowering could be ended when they just had fruits and no flowers left. And this is important because these plants are collected over a whole period of time. And if they're flowering for maybe a month between when the buds start to open till when they're in fruit, you want to be more precise and classify exactly what the plants are doing. So this is the specimens of Retibita columnifera. So we had 191 specimens from throughout Oklahoma. We just did the Oklahoma ones because we would have better data from there, with the different symbols being the different phases that the plants were in. And so we can look at the plants in these 4 phases by year. And so here we have the day of year, the day the specimen was collected on the horizontal axis, just like all our other graphs, and on the vertical axis, I'm sorry, and the horizontal axis is year of when the plant was collected. And we can see the lines are slightly sloped, but not very strongly, and the results were not statistically significant. So we couldn't tell that there was a trend in these data that was significantly different from no trend. We can't tell what's going on from this. But again, climate is variable, and so the year may not be the best way of looking at this if we want to see a response to climate. So we can look instead at the temperature of the year in which they are collected. And this is just for peak flowering. So here we have the day of year they were collected on the vertical axis, and then the statewide annual temperature. So the statewide temperature for that whole year on the horizontal axis, going from colder to the left and warmer to the right. And we can see that we do have some kind of a slope. Flowering was slightly, sorry, it should have been earlier in warmer years, but unfortunately not significant. But in addition to temperature being somewhat variable across years, temperature is also variable, and climate in general is also variable across the state. We know that the climate in the Panhandle is very different from the climate in the deciduous forests in eastern Oklahoma. And so it may make sense, instead of using one temperature for the whole state, to use the temperature of the specific climate division where the plant was actually found. And when we do that, this is what we get. We get a stronger and actually statistically significant relationship. So we have a downward slope, meaning that flowering is earlier in warmer years. And so we have found some relationship to our flowering. And when we look across all of our plants, 2 of them, we were not able to find significant relationships with temperature. So it may be that these plants are responding to day length. It may be that they are responding actually to rainfall or to something else entirely. But for these other 2 plants, with our Retibita, we had significant relationships between the date of flowering and the annual temperature of the climate division, as well as specifically the spring temperature of each climate division and summer temperature of the climate divisions. And then with the Grandelia, it was only sensitive to summer temperature, but either statewide or specifically of the climate division seemed to influence its flowering. So, and there are obviously lots more species of plants and lots more questions and analyses that can be done. But this is what John did for his research. So I would like to thank you for your attention, and I'm happy to answer questions now or later, whenever is best.
Oh, now would be good. We have one question, but I want to say thanks. This was a perfect follow-up to my very generalized bullet points. So this is like real research that you guys are doing on these exact questions and exact bullet points that I had mentioned in my very brief introduction. So it's cool to see the real data and what these studies are based on. And we do have one question from Zach. What kinds of herbarium specimens are worth collecting now? Should only records for like things new to the state or new county records be collected, or should people be collecting all sorts of things? As the herbarium curator, what's your feeling?
I say all sorts of things because, for example, if in order to do studies of the phenology of plants, what we really need is to have plants being continually collected. Like, we can't just have a record from 1950 and say, okay, we know everything about the flora. We've collected all these species in 1950, because then we wouldn't know how phenology changed. Like, the whole way you study phenology is by having this long record across all these different years. And so just because a plant has been collected in one place before, we need to collect it again, both to say that it's still there and to see what it's doing when it's flowering this year as opposed to the year that it was originally collected. So definitely valuable to keep collecting plants.
And I'd like to also add, as the invasive species person, that a lot of people have ignored things that are really common or what people consider as weedy. So, so those things are underrepresented in herbaria, which a lot of people wouldn't expect. You're like, oh, they're all over the place. We know all about those. But in herbaria, we don't have a lot of species specimens for a lot of what people consider common and weedy. So those are, although kind of boring and not very glamorous, that's important to add to our herbaria too.
Definitely. And the good thing with the weedy species is, especially if it's introduced, then you don't have to worry about destroying its population. You don't want to collect a rare native species if there aren't many individuals there, because they need all those individuals to reproduce.
Right. Grace has a question. How do phenophase categories change from herbs to trees? So I think probably she's asking, like, what kinds of life cycle things do you collect information on that's different from herbaceous things and trees?
So you can collect kind of these same categories with trees as well. In the trees generally flower, or I guess in the case of conifers, they produce pollen. Of course, people don't often, like oak trees or something, they often don't collect them when they're flowering. And so in addition with the trees, you can think about the time when the leaves are expanding and when the leaves drop and things like that. As far as herbarium specimens, you don't hardly get herbarium specimens of herbaceous plants when they're not flowering, because that's the only time you can identify them is when they're flowering. But with trees, you get a much wider variety because you can identify them at various times. Of course, if you have a specimen like this Quercus marylandica here, this blackjack oak, it's hard to say really, okay, it's fully leafed out, but it's fully leafed out for months. And so it's not that useful for phenological studies. The Rudbeckia hirta, the black-eyed Susan, is much more useful because it's only going to be flowering for a short period of time. But yes, you can definitely do phenology with trees, with herbarium specimens, and then we'll soon hear from Claudia about doing phenology of trees just with your observations.
We have one other question. Holly wants to know if you have enough fleabane in the herbarium collections.
We have lots of fleabanes in the herbarium collections, yes. There are several different species, quite a few different species, both in Oklahoma, and then especially as you go farther west, they get even more different species and it becomes very complicated.
Oh, that family is complicated. Every time I try to learn more about it, it's more complicated. Well, thank you, Abby, very much
But
for
it's
your
also very charming.
presentation. Oh, yes, yes. Thanks, Abby. I think that's the last question for now. Stick with us though, because we might have some more at the end, and then we'll switch it over to Claudia, who's going to talk about our community science project. Let's see, Claudia, can you switch over the
Hi.
screen to yours?
Yes. Alrighty. All right, can y'all see that okay?
Yes.
Okay, awesome. So I'm going to be talking about a phenology citizen science project that you can get involved in called Redbud Buddies. It's a group that we've created on a platform called Budburst. They have a website and an app. And if you have become more interested in phenology after this webinar, it's a really great way to get involved and learn more about the phenology of Oklahoma, native Oklahoma species, and in this case, the Eastern Redbud. So Budburst, it's a platform created by the Chicago Botanic Garden. It's a platform designed to connect researchers and members of communities interested in contributing to ongoing phenology projects. Budburst hosts a wide variety of projects that you can contribute to, one of those being observing phenology and climate change, others being observing pollinators and the relationship with pollinators and plants, and other plant and animal interactions. So if you'd like to participate, your job is to make observations on their app or website of a plant's life cycle. It's very similar to iNaturalist if you're familiar with it. And then you'd also want to provide details regarding your specific observation that are applicable to the phenology and climate change project. All right. So as I mentioned, they host a lot of different projects. However, the one that's most applicable currently is the Phenology and Climate Project, where researchers are aiming to answer the question, how is climate change affecting plant phenology? So I'm going to go step by step and show you how to create observations and how to join the Redbud Buddies group if you're interested. But the general overview, you just want to submit observations of Eastern Redbuds during spring, so now until June, via the Budburst app or website. You'll want to take photos of the plant and the tree, as well as recording phenological information, which I'll show later in a moment. and then you'll want to upload it to the 2022 Redbud Buddies group. So on this slide, I have a link to where you can register for an account, and then a photo of what the app looks like on your smartphone. I'll link any of the links that I have listed in this presentation to the chat once I'm done. And you can just search Budburst on the App Store of your smartphone. So once you open the app or get on the website, it'll bring you to these screens. I'll be showing kind of screen captions or screen captures of what it looks like on the app versus the website when going through this process of uploading an observation. So if you're on your smartphone, you'll want to click that button on the bottom that looks like a camera, and then you'll want to hit Start Observation on your website, on your web page. That would be the first thing that pops up when you log in and register. Afterwards, it'll open up your camera on your phone if you give it permission, and you can either take a photo right then and there in the app of the redbud that you're making your observation on, or you can choose an existing photo, or you don't even have to include a photo if you don't want to. It's very similar on the website. You can either upload a photo that you have on your computer, or you don't have to include a photo at all. You'll then want to search for the species that you are making an observation on. Very similar to iNaturalist. It'll give you suggestions based on the identification algorithm that the platform uses, but it's not always accurate. And with a project like Redbud Buddies, since you know the species that you're going to be making observations on, you can just quickly type that in and select Eastern Redbud. And then once you've selected the species you're making your observation on, it'll give you a little bit of an overview of information on the species, as well as some identification hints. You're going to want to hit Add Observation on your mobile device, or hit Next on your laptop or computer. And then the handy thing about the app is that it can take your location from where you are if you give it location privileges. So you don't have to worry about, typing in a specific address. I have blacked out my information because it's very accurate, which is handy. So you don't have to do anything on that page. On the phone app, for the website, you'll want to type in your address, or wherever the redbud that you're making an observation on is located. And you can be as specific as you feel comfortable. And then, like I mentioned earlier, Budburst offers a wide variety of projects that you can contribute to. However, for the Redbud Buddies project, we'd like you to contribute to the phenology project. So you'll want to click that option on your phone or computer. And then you'll also want to select the date that you made your observation on. And then, as I mentioned, you're going to want to add some phenophase information as well. So on the app and the website, they're going to ask you the same questions. And what you're going to want to do is click or tap those little circles within the larger circle about what specific information you're inputting. So for leaves unfolding, you'll want to look at the plant you're making an observation on, and just use your best judgment. They have some great guides on the Budburst website if you're unsure. And then as you're filling this out, once you click on any of the options, it'll give you a little sentence or two describing what to expect in that specific phase. So you're going to want to be filling out information on leaves unfolding, flowers, fruit phenophase information, as well as, although this isn't applicable at the moment because the Redbud Buddies project won't be active in the fall, but you'll want to record whether or not the leaves have changed color on the tree, or if any leaves are dropping. And then at the very end, before you submit your observation, you'll give you an opportunity to record any notes. It's totally optional. It's up to you whether or not you'd like to. And then you'll hit submit or save. So that's just kind of a general overview of how to submit an observation. You're more than welcome to participate in any of those projects that Budburst offers on your own in your own time. But if you would like to participate in the 2022 Redbud Buddies project, there's just one step you have to do before, which I'll walk you through now. So I've created a 2022 Redbud Buddies project on the app and the website. Once you have been added to this group, which I'll explain in just a moment, you're going to want to tap or click on that group specifically and then hit add observation. The way that the platform is set up, you're unable to, it doesn't automatically upload your observations into a group. Unlike iNaturalist, if you've participated in BioBlitz, you know that you don't have to specifically add to that project, and it just automatically does it if you're involved. So that's just something to remember if you'd like to participate. And then I just wanted to show you what a completed observation would look like. So in this, I created an observation on an Eastern redbud. I just grabbed a Google image and then selected, you know, what phenophase information I thought was applicable. And then in order to join the Redbud Buddies group, unfortunately, I do have to manually send out invitations. And because, you know, potentially quite a few people would want to join, I have created a survey where you can just send me your first and last name and your email that you've signed up with for Budburst, and I'll be able to manually send you invitations via the Budburst platform. So I will link this survey link in the chat of the Zoom, so you can, you know, save it or go ahead and fill it out if you'd like to participate. And if you have your smartphone with you, you can scan that QR code and it will take you to the survey as well. You're more than welcome to contact me if you have any troubles or any questions. Even though I'm, you know, not part of the Chicago Botanic Group, I can help troubleshoot as much as possible and answer any questions about participating in the 2022 Redbud Buddies group.
Thanks, Claudia. I also would like to add that I have added the information for the Redbud Buddies sign up. There's a link on our website now too. So if you miss that link or you miss it in the chat, there is a link on the website okbiodiversity.org now. We have one question from Zach, a general question for any of us. He's noticed that redbuds are beginning to flower earlier in town than in the nearby rural areas. Are there studies regarding the heat island impact on phenology? I'm sure there are. I don't know of any off the top of my head, but I'm sure somebody has been working on looking at the flowering occurring earlier in the heat islands. And it may be that we can take some of the phenological data that we're getting through Redbud Buddies or that gets submitted into the Botanic Garden Budburst program. And that should easily, since it's all geo-referenced, that should easily show some trends for heat islands, urban areas versus rural areas. Abby, did you want to add any information?
No, like you, I'm sure there are studies, but I'm not thinking of specific ones right now. But, but I am sure that has been shown because, yeah.
Yeah, I'm sure. I mean, just the fact that we have a good understanding about heat island, it would make sense that people are studying the ecology around that as well as just the meteorological stuff. Claudia's added the links to both the basic Budburst registration and then signing up for the Redbud Buddies program in the chat if you want to click on those and join us for spring to do your own phenological recordings. Let's see if anybody has any other questions. I don't see any new ones popping up. I guess since there aren't any new questions, we'll call it a night. Thank you all for joining us. Thanks to Abby and Claudia for presenting the information on our community science project and the research project going on in the herbarium. It's exciting to see research happening with these historic specimens that are collected in these natural history collections. So that's exciting. As exciting as dry plant presses press plants can be. So but it's important work that's getting done with these collections. Well, thanks everybody, and we will call it a night. I will announce all of you who registered will be added to our mailing list, and you will get an email when we announce our next webinar and the topic. So thank you all for joining us. I hope you have a good night, and start submitting those redbud observations. They're starting to bloom now.